Hey everybody, PreMedMama here. I'm a mother and a pre-medical student. Thank you for joining me. Maybe you're watching this on your phone or laptop or TV. Perhaps you're listening on your headphones, earbuds, or in your car. Whatever the case may be, wherever you are and whoever you are, I am so glad you're joining me. Let's get started. I'm in the Kaplan MCAT Biology review book. We're in Chapter 1, and today I'll be talking about Section 1-4, Genetics and Growth of Prokaryotic Cells. Open up your book or just keep listening. Here we go. As we have already explored, prokaryotic cells differ from eukaryotic cells, both structurally and biochemically. Prokaryotes reproduce via asexual reproduction in the form of binary fission. In addition, prokaryotes are capable of acquiring genetic material from outside the cell and using that genetic material. Binary fission. It's a simple form of asexual reproduction seen in prokaryotes. The circular chromosome attaches to the cell wall and replicates while the cell continues to grow in size. Eventually, the plasma membrane and cell wall begin to grow inward along the midline of the cell to produce two identical daughter cells. Because binary fission requires fewer events than mitosis, it can proceed more rapidly or more quickly. In fact, some strains of E. coli can replicate every 20 minutes under ideal growth conditions. At the end of this video, or below this video I should say, I'll be including some links to images and videos to supplement your understanding. Let's move now into genetic recombination. The single circular chromosome of a prokaryotic cell contains the information that is necessary for the cell to survive and reproduce. However, many bacteria also contain extra chromosomal or extragenomic material known as plasmids. Plasmids often carry genes that impart some benefit to the bacterium, such as antibiotic resistance, some mechanisms which are shown in a figure on page 22 in your book. Plasmids may also carry additional virulence factors or traits that increase how pathogenic or destructive a bacterium is, such as toxin production, projections that allow the bacterium to attach to certain kinds of cells, or evasion of the host's immune system. A subset of plasmids called episomes are capable of integrating into the genome of the bacterium. Bacterial genetic recombination helps increase bacterial diversity and thus permits the evolution of bacterial species over time. These recombination processes include transformation, conjugation, and transduction. Let's talk first about transformation. In a bacteria, transformation results from the integration of foreign genetic material into the host's genome. This foreign genetic material most frequently comes from other bacteria that, upon lysing or opening up, spill their contents in the vicinity of a bacterium that's capable of transformation. Many gram-negative rods are able to carry out this process. Next is conjugation. Conjugation is the bacterial form of mating or sexual reproduction. It involves two cells forming a conjugation bridge between them that allows for the transfer of genetic material. The transfer is unidirectional, or one direction only, from the male donor, positive, to the recipient, female, negative. The bridge is made from appendages called sex pili that are found on the donor male. To form the pilus, bacteria must contain plasmids known as sex factors that contain the necessary genes. The best studied sex factor is the F, or fertility factor, in E. coli. Bacteria possessing this plasmid are termed F+ cells. Those without it are called F- cells. During conjugation between an F+ and an F- cell, the F+ cell replicates its F factor and donates the copy to the recipient, converting it into an F+ cell. This enables the cell obtaining the new plasmid to then transfer copies to other cells. This method of genetic recombination allows for rapid acquisition of antibiotic resistance or virulence factors throughout a colony of bacteria, because other plasmids can also be passed through the conjugation bridge. The sex factor is a plasmid, but through processes such as transformation, which we just spoke about before conjugation, the plasmid can become integrated into the host genome. In this case, when conjugation occurs, the entire genome replicates because it now contains the sex factor. The donor cell will then attempt to transfer an entire copy of its genome into the recipient. However, the bridge usually breaks before the full DNA sequence can be moved. Cells that have undergone this change are referred to by the abbreviation capital H lowercase fr for high frequency of recombination. Now let's talk about transduction. That's the third recombination process for bacteria. Transduction is the only genetic recombination process that requires a vector, which is a virus that carries genetic material from one bacterium to another. Viruses are obligate intracellular pathogens. That's a term that we'll hear a lot more at the end of this chapter, which means that they cannot reproduce outside of a cell. Because of this, bacteriophages, which are viruses that infect bacteria, bacteriophages can accidentally trap a segment of host DNA during assembly. When the bacteriophage infects another bacterium, it can release this trapped DNA into the new host cell. This transferred DNA can then integrate into the genome, giving the new host additional genes. The process of transduction is shown in your book in Figure 1-13 on page 24. Again, I'll also include some additional resources underneath the video. Finally, transposons are genetic elements capable of inserting and removing themselves from the genome. This phenomenon is not limited to prokaryotes. It has been seen in eukaryotes as well. If a transposon is inserted within a coding region of a gene, that gene may be disrupted. Thanks for listening. Please tune in again soon. And if you liked what you heard, please subscribe to my channel and upvote my video. I believe in you. Thanks for your support and thanks for joining this community.